In this video lecture, I will be talking on adaptive or acquired immune system. Adaptive immune system reveals four characteristic attributes. These are antigenic specificity, which is a response to a particular antigen which is specific for that antigen. Diversity, the immune system recognizes different structures on non-self antigens on the surface of microbes. The adaptive immune system display immunological memory, so activated immune system upon first encounter will remember microorganism upon second encounter in a faster and more efficient way. The immune system or adaptive immune system has the capacity to discriminate between self and non-self antigens, and we mean by non-self foreign antigens. So the specific immune system normally distinguishes between self and non-self antigens and only reacts against non-self antigens. So cells of the adaptive immune system, all cells of the body, they come from stem cell, and cells of the adaptive immune system, which are B cell and T cell, they come from hematopoietic stem cell in the bone marrow. And this stem cell differentiate into self-renewal and differentiation cell, which further differentiate into myeloid progenitor and lymphoid progenitor. Lymphoid progenitor, they give cells of the adaptive immune system, which are B cell and T cell. B cells originate from bone marrow and they interact with the stromal cell of the bone marrow in presence of some cytokines, for example interleukin number 7, which helps in detaching of B cell from stromal cells, or precursor or progenitor B cell from the stromal cells following their interaction with this cell, where the B cell or progenitor B cell upregulate or regulate their membrane-bound immunoglobulin M, and they become immature B lymphocytes, which further regulate their membrane-bound IgD, and they become mature B cell, which exit from the bone marrow and patrol the body, and it reaches the secondary lymphoid tissues, the lymph node. Antigens that have the capacity to activate B lymphocytes, these could be thymus independent antigen or thymus dependent antigen. Thymus independent antigen are composed of repetitive identical epitopes on the surface of some microbe cross-link PCR. So these are the thymus independent antigen on the surface of some microbes, they cross-link the BCR, and this lead to independent activation of B lymphocyte in the absence of T lymphocyte. However, activation of B cell requires second signal, which is provided by Toll-like receptors on surface of B cell. So they express Toll-like receptor, which are known as pattern recognition receptors, and interact with pathogen associated molecular patterns on the surface of the pathogen. However, B cells differentiate into plasma cells, which produce IgM without producing memory cell. So in thymus or thymus independent antigens stimulate production of IgM, no class switch, no memory cell. That is to say, this results in production of plasma cells only. In case of thymus dependent activation, which requires T and B interaction, the antigen is usually protein, which has no repetitive epitopes. It is actually complex proteins. One of the most important cells of the adaptive immune system is T lymphocyte or T cell, which originate from bone marrow. But however, they migrate to the thymus gland for three purposes: education, maturation, development of repertoire, where they undergo positive and negative selection. Following their entry to the thymus gland through the high endothelial venules, they interact with thymic epithelial cells. Actually, they interact with the major histocompatibility class 1 and major histocompatibility class 2 expressed on the surface of thymic epithelial cells. So cells interact with major histocompatibility class 1 differentiate into CD8, and cells interact with major histocompatibility class 2 differentiate into CD4, and these are positively selected for the second step. The second step is that positively selected T cells, they go deep towards the medulla and interact with antigens expressed on the surface of antigen presenting cells present in the medulla, for example dendritic cells and macrophages. And if they fail to recognize this antigen, they will be killed by apoptosis, and this is known as negative selection. However, if also they recognize this, if they also recognize the antigen with high affinity, also they will be killed by apoptosis. Following that, they exit the thymus gland and patrol the body, where they reach the lymph node, and in the lymph node they interact with the B cell if they encounter an antigen. So T cell epitopes, TCR or T cells, usually recognize proteins, and T cell recognize primary protein. So as, as I mentioned before, that T cell is a blind cell. Blind cell means that they cannot see tertiary or quaternary proteins, but they see only primary protein. For that reason, tertiary proteins get phagocytosed and processed into primary proteins, which bind the major histocompatibility class 2 as a rough endoplasmic reticulum, and this primary protein or these peptides in association with major histocompatibility class 2, they get expressed on the surface of antigen presenting cell, so now T cells can see them. So T cells do not recognize polysaccharide or nucleic acid antigens, as these are recognized by B cell and known as T-independent antigens. Free peptides are not recognized by T cell unless they are presented in association with major histocompatibility, and this is T cell epitope. It possesses epitopes which binds the TCR, and agritope which binds the major histocompatibility cleft. So adaptive immune response requires cooperation between the adaptive or cells of the adaptive immune system and antigen presenting cells. The antigen presenting cells are dendritic cell, macrophages, and also B cell can act as an antigen presenting cell. So lymphocyte, they recognize antigen only in association with major histocompatibility expressed on the surface of antigen presenting cell, as can be seen here. These are antigen presenting cell. They present antigen in association with either major histocompatibility class 2 or major histocompatibility. This is in case of T helper 2 interaction with antigen presenting cell, or they recognize antigens, endogenous antigens, for example viral antigens or cancerous antigens, which are produced endogenously, and they bind the major histocompatibility class 1, and they get expressed on the surface, so they will be recognized by this is here the antigen presenting cell. They produce some endogenous antigens related to virus and to cancerous cell, and then they get coupled, or they bind major histocompatibility class 1, and they get expressed on the surface of antigen presenting cell. So peptides or endogenously produced antigens in association with major histocompatibility class 1 will be actually recognized by CD8. But however, here in this case, it shows T helper 1, T helper 2. T helper 2 actually likes to interact with B cell, while T helper 1, they like to interact with macrophage. So as I mentioned, that cells of the adaptive immune system, namely T cells, they do not see free peptides. They do not see tertiary proteins. They see only peptides in association with major compatibility, either major histocompatibility class 1, and these peptides presented in association with major histocompatibility class 1 will be seen by T cytotoxic or CD8, and this it shows major histocompatibility class 1, which is composed of alpha chain and beta 2 microglobulins. So TCR of T helper or CD4, they see processed peptides in association with major histocompatibility class 2, which has alpha and beta chain and express also a surface of antigen presenting cells. However, there is a third class of major histocompatibility. It is known as major histocompatibility class 3, which is this is encoded by genes present between the loci that encode major histocompatibility class 1 and major histocompatibility class 2, and they express CD4 or soluble substances, for example, CD4 toxic shock protein and tumor necrosis factor alpha. So major histocompatibility class 1 and 2 molecules are implicated in protection or susceptibility to disease, and also they are implicated in the development of autoimmune disease. Thank you very much, Prof.